Hello everyone, welcome to my YouTube channel. My name is Anthony Mogamedio, a student of Biomedical Level 3 from Group 5. I'm going to base on question 6, 7, 8, 9, 10 on the topic of applied immunology. Question 6 says, how does central tolerance prevent autoimmunity? Central tolerance is a critical mechanism in the immune system that prevents autoimmunity by ensuring that the developing immune cells, particularly the T cells and the B cells, do not attack the body's own tissues. This process occurs in the primary lymphoid organs such as the thymus for the T cells and the bone marrow for the B cells. And it's working like in a two-way, that is a negative selection of self-reactive, that is the T cell and the B cells, and then the positive selection of non-reactive cells. Allow me to talk about the positive selection for non-reactive cells. In T cells, the thymocytes must demonstrate a moderate ability to bind to self-MHC molecules to survive, ensuring they can recognize the MHC when presented with foreign antigens. In the peripheral cells that fail, these tests also undergo apoptosis. What is autoimmunity itself? Autoimmunity is a condition in which the immune system mistakenly identifies the body's own cells, tissues, or organs as foreign, attacks them. Normally, the immune system defines the body against harmful pathogens like bacteria, viruses, while recognizing and tolerating the body's own tissue. Self-tolerance. In autoimmunity, this tolerance breaks down, leading to immune responses against self-antigens, resulting in the tissue inflammation, damage, and dysfunction. Enumerate 12 main medically important autoimmune diseases, their causes, and their clinical manifestation. Let me first go with rheumatoid arthritis. The cause is unknown, but it is believed to involve genetic predisposition, environmental factors such as smoking and hormonal influence. The clinical manifestation of RA is systemical joint pain, swelling, morning stiffness lasting more than an hour, fatigue, fever, loss of appetite. Over time, it can lead to joint deformities. We go also with type 1 diabetes mellitus. We all know diabetes mellitus is an autoimmunity destruction of insulin-producing beta cells in the pancreas. Genetics predisposition combined with environmental triggers like viral infection may play a role. The clinical manifestation is we have the increased thirst, urination, extreme hunger, that is polyphagia, weight loss despite eating well, fatigue, blue vision. Long-term complications include cardiovascular disease and neuropathic. Graves' disease causes is being caused by autoimmune disorders that leads to overactivity of the thyroid gland, though to be influenced by genetic factors. Stress. The clinical manifestation of Graves' disease is hyperthyroidism symptoms including weight loss despite increased appetite, rapid heartbeat, heart tolerance, anxiety or irritability may also cause blooding eyes. Let's go to question 7. What immune checkpoints and why are they important? Why are they important? What are immune checkpoints? We all know immune checkpoints are regulatory pathways within the immune system that control the intensity and the duration of immune responses. These checkpoints act as molecular brakes to prevent overactivation of the immune system which could damage healthy tissues. They play a crucial role in maintaining immune balance, self-tolerance, preventing autoimmunity disease, and limiting tissue damage during immune responses. Immune checkpoints are mediated by receptor-ligand interactions between the immune cells, primarily involving T cells and antigen-presenting cells. Why are they important? Checkpoints. Why immune checkpoints are also important? One, it prevents autoimmunity. Two, minimization of tissue damage. Three, regulation of chronic immune responses. How? They play a role in deepening immune responses during prolonged infection or inflammation, preventing excess of immune cells, and also play a role in cancer. Many tumors exploit immune checkpoints like PD-L1 and PD-1 to evade immune detection effectively, hiding from the T cells. This is why immune checkpoint inhibitors have become a major focus in cancer immunotherapy. How do the cytokines regulate immune system? How do cytokines regulate immune system? Cytokines are small signaling proteins secreted by immune and non-immune cells that play a central role in regulating the immune response. They act as messengers between cells, overstating the development, activation, and resolution of immune reaction. Their functions: activation of immune cells. How are they activating the immune cells? Cytokines stimulate the activation and proliferations of immune cells. Interleukin, that is 2IL-2, it promotes the T cell proliferations and survival. Granulocytes, macrophages, colony stimulating factor. It stimulates the production of neutrophil, macrophages, and dendritic cells. Two, is also help in differentiation of helper T cells, and it's also help in promotion of inflammation. It's also resolution of inflammation, and is coordination between the innate and adaptive immunity. Cytokines, the gap between the innate and adaptive immunity, are the interferons produced during viral infection to activate natural killer cells and enhance antigen presentation to T cells. And we also have a cytokine storm regulation. Let's go to question 9. That's how do monoclonal antibodies work in immunotherapy? Monoclonal antibodies are laboratory engineered antibodies designed to target specific molecules, that is, antigens involved in disease process. In immunotherapy, they help modulate the immune system or directly attack diseases causing cells such as cancer cells, infections, agents, autoreactive immune cells. Mechanisms of action of monoclonal antibodies immunotherapy. One, they are targeting and marketing cells for destruction. MABs can bind to specific antigens on disease cells. Example, like the cancer, they can bind with, marking them for destruction by immune system. Antibody-dependent cellular cytotoxicity. Natural killer cells bind to Fc region of the antibody and kill the target cell. And CDC binding of antibodies activates complement cascade leading to cell lysis. Example, we have in the rituximab an anti-CD20 and it targets the B cells in non-Hodgkin's lymphoma and autoimmune disease. 3 is the block growth signals, immune checkpoint inhibition, and lastly we have immune cells system modulation. Here, some MABs modulate the immune responses by targeting cytokines or their receptors, reducing inflammation in autoimmune disease. Example, we have in the adalimumab, we have in the torisizumab, and so many others. What are the advantages? One is specificity, highly specific for their targets, reducing off-target effects. Versatility can be tailored to virus diseases including cancer, autoimmune disorder, and infection disease. And 3, combination therapy. This can be used with other treatment like chemotherapy, radiation, or other immunotherapies. Let's go to question 10. What is the principle behind checkpoint inhibitor therapy? Principle behind checkpoint inhibitor therapy. Checkpoint inhibitor therapy is based on the principles of blocking immune checkpoint pathways to enhance the body immune system to particularly against cancer cells. Immune checkpoints are natural regulatory mechanisms in the immune system that act as brakes to prevent excessive activation. Tumors exploit these pathways to invade immune depletion and destruction. Checkpoint inhibitors are molecular antibodies designed to block these inhibitors pathways, effectively releasing the brakes, allowing the T cells to attack the tumor cells more effectively. So when we release the brakes here, it makes the T cells to attack the tumor cells automatically. The key immune checkpoint targets in therapy were having the PD-1 and PD-L1 pathway, program death, that is the meaning of PD, program dead one, a receptor of T cells that dampens T cell activity when bound to its ligand, PD-L1 or PD-L2. PD-L1 often overexpressed on tumor cells, allowing them to invade immune attack. Checkpoint inhibitor mechanisms. Drugs like nivolumab and pembrolizumab block PD-1, preventing its interaction with PD-L1 and PD-L2. This restores the T cell activity and enables them to attack the tumor. CD-LA4 pathway, cytotoxic T lymphocyte associated protein, a receptor on T cells that inhibits the activation by outcompeting the co-stimulatory receptor. CD28 for binding to B7 molecules on antigen presenting cells APCLs. Checkpoint inhibitor mechanism. Drugs like ipilimumab block CTLA4, enhancing the T cells priming and activation of lymph nodes. This amplifies the immune responses against tumors. Steps in checkpoint inhibitor therapy. Immune invasion by tumors. Tumors exploit immune checkpoints like PD-1 and PD-L1, CTLA4 to suppress the T cell activity and invade immune destruction. Checkpoint blockage. Checkpoint blockade inhibitors bind to PD-1 and PD-L1 acetyl-4 to block their interaction. This prevents tumor-induced suppression of T cells. Number three, we're having the T cell reactivation. Blocking these pathways allows the T cell to remain active, produce and target tumor cell effectively. Applications of checkpoint inhibitor therapy. Checkpoint inhibitors have shown remarkable success in treating a variety of cancers, that is including the myeloma, non-small cell lung cancer, renal cell carcinoma, and Hodgkin lymphoma, head and neck cancer. Thanks for listening. This was my presentation based on applied immunology from group 5. So see you again in the next presentation. Thanks.